How can enzymes be so precise in such a messy, crowded cell? This is the heart of the enzyme specificity paradox. Enzymes somehow select one perfect molecule from thousands of similar ones around them. Are they just rigid locks waiting for the right key? Not quite. While early models suggested that enzyme active sites are pre-shaped to match specific targets, this does not explain how they ignore so many lookalikes. So what is the secret? It turns out enzymes are flexible. They shift shape to mold around their true target, a process called induced fit. But even that is not the whole story. How can they tell apart molecules that differ by just a hydrogen atom or that are mirror images? The answer lies in their ability to detect subtle features, like electric charges, hydrogen bonding patterns, and even fleeting quantum-level interactions. Some enzymes are also designed to be versatile, able to handle multiple substrates when needed. Can we control this? Yes. Scientists now tweak enzymes using directed evolution, reshaping their preferences in the lab. So what does this all mean? It shows that enzyme specificity is not just about fit. It is a delicate, dynamic process of sensing, adjusting, and responding. In a sea of noise, enzymes find their perfect signal, reminding us how biological precision can arise from elegance, not complexity.
At first glance, it seems impossible. Inside a living cell, a chaotic soup of thousands of molecules bumping, folding, twisting, and reacting every second, an enzyme is expected to find one specific molecule and catalyze one specific reaction, and yet it does. Not just once, but millions of times per second with mind-blowing precision. Even when there are dozens of similar-looking molecules nearby, the enzyme almost never makes a mistake. How is that possible? This is the heart of the enzyme specificity paradox. How can enzymes be so exquisitely selective in such a crowded, noisy, molecular world? To understand the paradox, let's start with what enzymes are. Enzymes are biological catalysts, proteins that speed up chemical reactions without being consumed. Each one is tailored to perform a very specific task, like cutting a sugar molecule, assembling a protein chain, or copying DNA. They're nature's precision tools, but the level of precision they display, often called substrate specificity, isn't just high, it's astounding. Imagine being blindfolded in a room filled with thousands of keys, all jumbled together. You're asked to find the one key that fits a particular lock and insert it at the exact right angle to open it. Now imagine doing that 10,000 times per second without making a mistake. That's essentially what enzymes do every moment in every cell of your body. So how do they do it? The classical explanation is called the lock and key model. In this view, the enzyme has an active site, a uniquely shaped pocket that only one specific molecule, the substrate, can fit into, like a key fitting into a lock. If the fit is wrong, even by a tiny molecular edge, the reaction won't happen. This explains part of the story, but it raises a deeper question. What if other molecules are extremely similar in shape or size? Why don't they also fit the lock? Here's where the paradox sharpens. In many cases, the molecules surrounding an enzyme differ from its true substrate by just a single atom or bond, yet the enzyme still ignores them. It's like distinguishing between identical twins based on a slightly different hairstyle while wearing a blindfold and moving at light speed. To solve this, biochemists propose a more dynamic idea, the induced fit model. Instead of being a rigid lock, the enzyme is flexible. When the correct substrate approaches, the enzyme changes shape slightly to form a tighter, more perfect fit, like a handshake adjusting to the person you're meeting. This extra level of interaction adds conformational precision, allowing the enzyme to feel out subtle chemical differences and reject imposters. Still, that's not the whole picture. Enzymes don't just recognize their substrates, they often bind them in a specific orientation, position key amino acids to stabilize the transition state, and even exclude water molecules or competing ions from the active site. These mechanisms act like molecular gatekeeping, ensuring that only the correct substrate in the correct state triggers the catalytic reaction. You might ask, can enzymes ever make mistakes? Yes, but rarely. Some enzymes have error rates of just 1 in a million, and even when errors do occur, cells have repair mechanisms to catch them. In DNA replication, for example, The enzyme DNA polymerase uses multiple layers of specificity, including proofreading steps, to ensure nearly perfect fidelity. And this is all happening inside a cell at body temperature without any external sensors or computers. One reason enzymes can be this precise is that evolution has optimized them over billions of years. The amino acids that form the active site are carefully arranged, not just for binding, but for excluding anything that doesn't fit exactly. In some cases, a single change in pH, temperature, or structure can collapse this whole system, leading to loss of function or disease. But perhaps the most astonishing examples of specificity come from artificial enzyme design. Scientists can now engineer or evolve new enzymes in the lab, and sometimes these synthetic catalysts show selectivity for molecules that nature has never seen before. Yet even here, the underlying challenge remains, distinguishing one molecule from a sea of lookalikes, using only molecular interactions and folding patterns. So what's the deeper meaning of the enzyme specificity paradox? It reminds us that precision in biology doesn't require complexity, it requires finely tuned interactions. At the nanoscale, even weak forces like hydrogen bonds or van der Waals interactions can create powerful selectivity when arranged correctly. It's not brute force that makes enzymes work, but elegance, a choreography of shapes, charges, and movements that filters the world at atomic resolution. In a universe governed by statistical noise and thermal chaos, enzymes are islands of order, proving that life has found a way not only to function, but to do so with surgical precision. And perhaps most remarkably, they show that even in the messiest environments, clarity can emerge from chaos. Guided by the right molecular questions, the enzyme specificity paradox refers to the astonishing ability of enzymes to discriminate between highly similar molecules and catalyze reactions with exceptional speed and accuracy, often under complex cellular conditions full of noise and competing substrates. This paradox challenges the notion that biological environments are too messy for such precision. Instead, enzymes achieve selectivity levels that often surpass synthetic catalysts, raising deep questions about the molecular logic and design principles underlying biological recognition and catalysis. DNA polymerase can differentiate between the correct and incorrect nucleotide with an error rate of less than 1 in 107 insertions, despite all nucleotides being chemically similar. It's like a vending machine that in a dark room can always find the exact coin you tossed in, even if dozens of others are nearly identical in size and weight. Substrate specificity is the enzyme's ability to selectively bind and transform one molecule, the substrate, out of a pool of similar candidates. This specificity arises not from a single factor, but from a multi-dimensional recognition process involving shape, steric fit, charge complementarity, hydrophobic interactions, and dynamic conformational changes. The classic lock and key and induced fit models only partially explain the exquisite discrimination. Modern understanding emphasizes dynamic, adaptive binding sites. Trypsin cleaves peptide bonds after lysine or arginine residues, ignoring other nearby amino acids due to its deeply positioned, negatively charged pocket that matches only basic side chains. It's like a glove that doesn't just fit one finger size, it reshapes slightly to tightly embrace one particular finger, rejecting even close lookalikes. Enzyme specificity is achieved through cooperative molecular mechanisms, including transition state stabilization. Enzymes bind most strongly not to the substrate itself, but to the high-energy intermediate formed during the reaction. Energetic checkpoints binding often involves multiple reversible steps, creating a funnel that favors the correct substrate. Kinetic proofreading, some enzymes expend energy, e.g., via ATP hydrolysis, to check substrate identity multiple times before committing to catalysis. These mechanisms create extremely steep energy landscapes, where even small changes in molecular geometry or charge drastically reduce binding affinity or reaction rate. Amino-acyl tRNA synthesis charge tRNAs with their corresponding amino acids, rejecting incorrect substrates through a double sieve mechanism, first by size, then by chemical reactivity. It's like an airport security system with two checkpoints. Only the correct passenger with matching passport and boarding pass gets through, even if others look nearly identical. The enzyme specificity paradox showcases nature's ability to resolve complexity into order at the molecular level. Despite molecular crowding, thermal motion, and competing interactions in the cell, enzymes function with clockwork precision. This illustrates the principles of emergent selectivity, where system-level accuracy arises from local, dynamic interaction. Enzymes act not as rigid structures, but as adaptive molecular machines that process chemical information and respond to environmental cues. In metabolic networks, enzymes maintain fidelity even when intermediates are reused in multiple pathways, thanks to localized compartmentalization and sequential binding cues. It's like finding your way through a busy city using not just GPS but also contextual signs, traffic lights, and local landmarks to stay precisely on course. High specificity often involves energy input either directly through ATP/GTP hydrolysis or indirectly via binding energy. This energy is used to amplify tiny differences between similar molecules and prevent incorrect reactions. Some enzymes couple exergonic reactions to drive conformational changes that lock in the correct substrate or eject incorrect ones. These cycles reflect a thermodynamic trade-off. Accuracy costs energy but ensures fidelity in critical processes like DNA replication or protein synthesis. The proofreading subunit of DNA polymerase removes mispaired nucleotides by using energy from the phosphate backbone cleavage, ensuring genomic stability. It's like checking a document twice before printing, burning some ink and paper in the process, but avoiding a costly error. From an information theoretic view, enzyme specificity is about maximizing mutual information between molecular identity and catalytic outcome. The enzyme acts as a decoder that extracts meaningful input, the correct substrate, from noisy biochemical environments. Selectivity reduces entropy and increases information fidelity, analogous to how a neural network filters out noise to recognize a face or voice. Binding affinity, kinetic barriers, and conformational matching serve as error correction mechanisms. The ribosome ensures accurate translation by favoring codon-anticodon pairings that match Watson-Crick rules, using energetics and shape complementarity as discrimination filters. It's like a voice recognition AI that recognizes a speaker not only by words but by pitch, rhythm, and emotional tone, all signals working together to reduce uncertainty. Understanding enzyme specificity has far-reaching applications, from drug design and synthetic biology to biosensing and industrial catalysis. Drugs can be engineered to mimic transition states, inhibit active sites, or exploit off-tagment effects. In synthetic biology, custom enzymes are being evolved to accept novel substrates. Artificial intelligence systems are learning from enzymatic logic to develop pattern recognition algorithms in chemistry and materials science. HIV protease inhibitors mimic the transition state of peptide cleavage, fitting the active site with nanometer precision to block viral replication. It's like crafting a master key that fits a lock so well it freezes the whole mechanism, stopping it from working for anything else. Despite advances, predicting or designing enzyme specificity remains a grand challenge. Small sequence changes can lead to large, unpredictable shifts in specificity. Enzymes often display promiscuity, catalyzing secondary reactions that complicate engineering. Modeling specificity requires high-resolution structural data, molecular dynamics simulations, and AI-guided prediction tools, yet emergent behavior from protein flexibility remains hard to capture. Attempts to design synthetic enzymes for Diels-Alder reactions succeeded only after multiple iterations of directed evolution and structure-guided mutagenesis. It's like designing a musical instrument that can only play one perfect note in a storm. Too many vibrations and echoes can throw off the tune. Future research will focus on programmable enzymes, allosteric control mechanisms, and adaptive catalysts that change specificity in response to external signals. Hybrid enzyme-AI systems may enable real-time redesign of catalytic preferences for personalized medicine or adaptive manufacturing. Enzymatic specificity also serves as a model for intelligent filtering systems. From neuromorphic computing to autonomous chemical robots capable of molecular decision-making, CRISPR-associated enzymes now offer programmable DNA targeting based on nucleotide sequence recognition, a new frontier in enzyme specificity and synthetic gene control. It's like teaching a dog to sniff out only one scent in a forest, then reprogramming it to detect another using nothing but a few molecular cues.